Okay, so in this section of material, we're going to be looking at 9.3 evolution of populations, and we have a nice little comic here looking at how Twitter is upgrading our gene pool. When we look at evolution on a micro scale, we're really looking at how the alleles frequencies change within that population. That's a lot different than what most people would consider or think of when we speak of evolution. A lot of times they think of things along the lines of macroevolution, where we're seeing emergence of brand new species. That process takes a lot longer to happen, but microevolution is something that we can witness and kind of keep track of even during our own lifespans, looking at data, either historical data from our own human population or maybe a population of another organism that has a shorter lifespan, or we have historical data to look back through. All right, so really what we're talking about is genes and variation, and we should keep in mind that all alleles within a population are considered the gene pool, and the relative frequency is going to be the number of times that allele actually occurs within that gene pool. It's usually expressed as some percentile number, so like an amount out of the total number of alleles present. So in this illustration, we have a pond of frogs, and we have big G coding for the green color, little g codes for brown. And so if we look at our gene pool here, as far as like each individual, we have some green individuals that are big G, we have some green individuals that are big G, and then we have our homozygous recessive brown frogs, little g. And so we can calculate the actual allele frequencies for each allele present. And if we count these guys up, we see that there are 7 big G's in our gene pool to 5 little g's in our gene pool. So that means that we have a total of 12 alleles present for skin color in this pond. And the frequency of the dominant allele would be 58.3%, is 7 divided by 12. And the frequency of the recessive allele, 5 over 12, or the 41.7%. It would make up the other portion of the 100%. So if brown skin color became advantageous, what would happen to these allele frequencies of big G and little g? Well, if brown skin was actually going to allow you to procreate at a higher success rate, then we would expect that the small recessive allele to increase in its frequency, that we would see it occur more often because little g would be being passed on to subsequent offspring. All right, remember that these allele frequencies should add up to 100%. So if we consider this white versus black alleles, if you have 2 black, then we have a black mouse. If we have 2 whites, then it's a white color mouse. So black is the dominant allele. And the frequency in this population of mice would be 12 out of 20. We have 10 individuals, 2 alleles each, and so 20 alleles to consider. And the frequency of white would be 8 out of 20, or 40%. So the question is, does gene frequency equal phenotype frequency? Right? Notice that we have 60% black allele, but if we look at our numbers here, we have 1, 2, 3, 4, 5, 6, 7, 8. 8 of our 10 mice are actually black in fur. So that would be an 80% occurrence of the phenotype. So, why is that? Well, they don't match because some black mice will carry that recessive allele. Even though they have the white recessive allele, the dominant gene is what is expressed or what overshadows the other trait. All right, so with population genetics, then we simply look at combining Darwinian selection and Mendelian's idea of inheritance together. So it's a study of genetic variation within that population, and we really emphasize quantitative numbers or characters. Remember that evolution is going to act on populations, not individuals. So the population can be defined as individuals of that same species within a given area. So all members of a population can interbreed, right? It's a possibility. And so they share a gene pool, a set of alleles that are present for all traits within that population. All right, now, if all members of a population are homozygous for a certain allele, then we would say that that allele is fixed in the gene pool because there's no option for having the other recessive or dominant trait, whatever we happen to be homozygous for. Usually, more than 2 alleles for a gene are going to occur in a population, and so each will have a relative frequency or proportion within that gene pool. So how can we determine when the population is actually evolving? Well, we use a theorem known as the Hardy-Weinberg theorem. Hardy-Weinberg it works when you consider evolutionary pressures not to be present, right? So we can look at a non-evolving population where the gene pool remains constant over multiple generations. So the allele frequency does not change over generations of time. Natural populations, we know, are not expected to actually be in Hardy-Weinberg equilibrium, right? We know that microevolution is occurring when those allele frequencies do in fact change. So the deviation from this Hardy-Weinberg equilibrium usually results in evolution in that population. So really, if we understand a non-evolving population, we can then identify the conditions in which a population is in fact going through that evolution. There are some assumptions for a population to be in Hardy-Weinberg equilibrium. All right. So first thing is it's a very large population size, sometimes referred to as an infinite population, because if we have a small population, a small change in allele frequencies could make a huge effect on the percentages. So we consider a very large population. We also consider there's no migration, because if we have things coming in or leaving our population, then they could bring different alleles into the population that were not there before, or we could lose alleles that were once present through migration. So we assume no migration. We assume no net mutations, because again, if we have a mutation in the code, then that could result in a new allele that was not present before, or remove one that we had. Random mating, that there's no trait that's more desirable than in another. We know naturally this is not true, that certain traits are usually more successful for procreation than others. And if that was occurring, if it was not random, then there'd be a definite favored set of traits that would be more successful. Remember that assumptions about the Hardy-Weinberg theorem is that evolution is not occurring, so you can try to predict or determine allele frequencies over time. And then no natural selection, that we do not have traits that are more advantageous than others to allow that individual to survive more successfully than another one. And again, in green, we mentioned what's actually happening in reality. We know that some individuals will survive and reproduce better than others. And so in Hardy-Weinberg's theorem, he makes the assumption that none of this is going on. All right, so the Hardy-Weinberg equation is actually not too difficult. What most people run into a problem with is what the squared variables actually represent. It's where 1 is equal to p squared plus 2pq plus q squared. Now, for organisms, p2 or p squared represents the frequency of the homozygous dominant genotype. Right? So whatever p squared is, it equals the frequency of the homozygous dominant genotype. 2pq is representing the frequency of the heterozygous genotype, big A, little a in this example. And then q squared is actually representing the frequency of the homozygous recessive genotype. The key takeaway here is that if you know one of them, if you know what p squared is equal to, you can square root it, and that's going to give you the percent of that allele that's present compared to the recessive allele. And we can actually figure out what q or the other one might be by remembering that 1 is equal to p plus q. Right? Remember, p by itself is just showing us the percent of the alleles that are the dominant type compared to the percent that are the recessive type. Just like we did in our frog example a few slides back. All right, so for alleles, then again, the little p is frequency of dominant allele, like big A, and q is the frequency of the recessive allele, little a. Okay, so how might this be useful? How might we use this? Well, we can actually, there's a gene that actually codes for the ability to test PTC, which is a chemical that can be found in some foods, and it tastes very, very, very bitter, right? On average, though, 75% of people can taste it, while only 25% usually cannot. And really, it depends on, if we look through the information here, and this is extra information for you to kind of check out, the ability is conveyed by a single gene that codes for a taste receptor on the tongue. So, one of the common forms is a tasting allele. The other is a non-tasting allele, right? And each allele codes for the bitter taste receptor protein with a slightly different shape. So the shape of the receptor protein determines how strongly it can actually bind to PTC and actually detect that it's there. So since all people have 2 copies of every gene, because they have one from mom, one from dad, the homologous pair, the combinations of that bitter taste gene will vary how intense that taste is. All right, some will actually absolutely detect a very bitter taste if they have both strong variants. If they have one strong and one weak, one from mom, one from dad, or vice versa, they'll be somewhat bitter. And if they both have the non-tasting alleles, then they wouldn't really detect a bitter taste at all. So we know that PTC is not actually found naturally, but it does correlate strongly with other tastes of bitterness and other substances that do naturally occur. Most of them, of course, are toxins that our body's trying to warn us about what not to eat. But some people think that this might have something to do with whether or not people would actually find smoking more pleasurable than others, because some people would really pick up on that really bitter taste of the smoke from doing that, or maybe even like whether they like broccoli or not. That this ability to taste things that are rather bitter, and if there's a really strong taste to them, that would turn you off to eating that vegetable. So, what if we were to try to determine some things about in a Hardy-Weinberg sort of way, using the Hardy-Weinberg equation, as far as like the frequencies of the different phenotypes in a population? All right, so we're going to use this idea of the PTC taste ability, and this information was looked at in a Caucasian population where 70% of people can taste it, 30% cannot. And if we look at the overall population statistics, it comes out to about 75% of the overall population can actually taste PTC. All right, so let's hop out of this and let's do a little bit of work with the Hardy-Weinberg equation. All right, so we'll work with what's actually in the practice problem here, right? But you can always redo this problem by looking at the overall statistics for the entire population. All right, so first things first. They want us to determine the frequency of the homozygous recessive phenotype, or Q squared. Remember, our Hardy-Weinberg equation is that 1 is equal to P squared plus 2PQ plus Q squared. Okay, so if we want to know what the frequency of the homozygous recessive phenotype is, then we're told that 30% are non-taste testers, or non-testers, tasters for the bitter taste. So they would be at a 30% rate as far as the recessive phenotype, right? So if they're non-tasting, they must be like, we'll say little a, little a. Okay, so they can't taste this. And so this is going to be at 30%, or we can put 0.3. Should be equivalent to 30%. All right, so now that they want us to find out the frequency of the recessive allele, all right, so this is going to be like how often in the population do we see or do we have little a happening? Now, from this equation, we're told that Q squared is this 30%. Right, so in order to determine the frequency of the recessive allele on its own, we would want to square root the 30%. Right, so we would want to take this, because this we're told, if we scroll down a little bit, we pull this out of the equation, we're really saying that Q squared, whatever it is equal to that 30%. 0.30. So in order to solve for whatever Q might be, which is the frequency of the recessive allele, we're going to have to square root the 0.3, right? Because we have to square root on the left-hand side to get rid of the square. So whatever we do to the left-hand side, we have to do to the right-hand side. And so the square root of 0.30 actually ends up being 0.5477. 0.5477. So that's going to be the frequency of the recessive allele, 0.5477. Or around 54.8% if we were to move it to a full number percent. All right, so then they're asking us for the frequency of the dominant allele. Okay, so remember, not only do we have Hardy-Weinberg's equation here, right? Not only do we have this, but we also know, if we extend the page down, we're also going to have been told that 1 is equal to P plus Q. So that means 1 is equal to whatever P is plus our 0.5477. That means P has to be equal to, well, it's going to be 1 minus our 0.5477, or 0.4522, or sorry, 0.4523. So we can hop back up, 0.4523. Notice it's really easy because these guys have to equal, these guys right here have to add up to 1. Right? What about the frequency for homozygous tasters? Well, that's just P squared. So we know what P is. It's 0.4523. So we're going to simply take our 0.4523 and we're going to, you guessed it, square it. And that's going to give us the frequency of the homozygous tasters. Or roughly 20.5% or so. All right, and then as far as the heterozygous tasters, we're going to make use again of that Hardy-Weinberg equation, noting that we're now after this 2PQ. Right, so we had to set up our math here. I'm going to clear a little bit off this board if I can. So we know that it's going to be equal to 2 times P, right, which is 0.4523 multiplied by Q, which is 0.5477. And if we run this through our calculator, we're going to find that this is going to be equal to 0.4953. And we'll put that in up here, 0.4953, which would be about 49.5% or so. Right, which we would expect, right? The homozygous recessive phenotype is around 30%. The homozygous tasters are at 20.5%, and the heterozygous tasters are at 49.53%. If we would add this percentage to this percentage to this percentage, then it adds up to 100%, or adds up to 1. So that's a quick way to kind of double check to make sure that we've used this Hardy-Weinberg equation appropriately. So what I might suggest is try this again, but go with the 75% being able to test or taste, and then with 25% not, and see how that turns out for you. Okay, so if we look at parent populations from maybe flower color, right? So we have genotypes of big R being the dominant red color and little r being the recessive white color. And we have 320 that are big R. We have 160 that are heterozygous and 20 that are homozygous recessive. If we run these numbers, we can find the genotype frequencies
for the homozygous dominant and the homozygous recessive, and as well as the heterozygous. And these values, this 0.64, would be equal to p squared, right? And this big R little r heterozygous would be equal to 2pq, and this 0.04 would be equivalent to q squared. So if we then flow through the individual alleles down to the allele frequencies, we can see that it ends up coming out to, as what we expect for p, the square root of 0.64 would be 0.8. And so we have 80% frequency of the dominant allele and a 20% or 0.2 frequency of the recessive allele. We can also then apply this to a Punnett square, and that's basically where we why we use the Punnett square to see these percentages of occurrence. And we would expect in this generation then for the genotype frequencies to end up playing out with p squared being 0.64, 2pq being 0.32, and that would be the pq here in the left or rightmost square plus QP in the leftmost square. So add those together, we have 0.32, and then our recessive white flower would be the 0.04 frequency. So another thing we need to touch on here is evolution. Evolution within a species or population is known as microevolution, and that's where we see an actual change in the allele frequencies. This will happen if we do not consider the population to be not at Hardy-Weinberg equilibrium. So this is where those causes of microevolution would come into play, like genetic drift, meaning like, especially in small populations, as certain traits are more advantaged than others as far as the reproduction of that species or of that population. Natural selection pressures, gene flow, mutation, and also non-random mating, where certain traits are advantageous for finding a mate or actually mating with an individual. In terms of variations between populations, we have geographic variations where there's differences between the gene pools because of environmental factors like cline. Cline is where by different elevations we find different types, or climates, we find different types of organisms present. And so insects and birds are larger in warmer climates than those we find in cooler climates. And maybe this is because smaller body sizes help conserve the heat better. So if you're like in a cooler location, you would want to be a smaller organism. Another client example would be maybe heights of these yarrow plants, right? And so we have the Sierra Nevada Range and the Great Basin Plateau after the elevation increase, and we find that as we go along, as we go up in elevation, we find that the plants don't grow as tall or as big because, again, we're looking at a more limited set of resources that are available as we go up in elevation. Polymorphism is where we have two or more forms of a character. These are morphs, so poly would be many morphs in high frequencies within a population. So this applies only to discrete characteristics like the jaguar coloration, or another example would be sexual dimorphism, because there's distinct differences or appearances between male and female sexes of an animal, in addition to differences between the actual sexual organs themselves. So even like that, men on average tend to be a little taller than women. The actual shape of the individual also. So differences in size, height, structure, appearance can also be attributed to certain sexes, either male or female. Diploidy and balanced polymorphism really try to help preserve variation. We know that diploidy or diploidy is where we have more than one allele for a trait, and so we could have dominant and recessive forms of that trait, and they help hide genetic variation from selection in the form of those recessive alleles, that if you have a heterozygous individual, the dominant trait will show, but they're still carrying with them unknowingly or unknown to maybe others, that recessive allele. So they kind of hide that recessiveness. The heterozygote advantage then is where the heterozygote actually has a greater chance of survival and reproductive success than either homozygote. Maybe like sickle cell anemia, where the heterozygotes, big A, little a, negative, are resistant to malaria. But if you're homozygous dominant, you get malaria, and homozygous recessive has sickle cell disease. And this is like a distribution of frequencies of sickle cell allele as they occur, and then the distribution of malaria caused by this protozoan. And so we can see that the frequency of the sickle cell allele is present in higher degree where malaria tends to happen in higher occurrence. All right, so neutral variation is where the variation actually results in no competitive advantage to either individual, and so human fingerprints would be one of these variations where there's really no gain or loss to having unique fingerprints. Now, the last thing to kind of mention here is that sexual selection really does lead to differences between the sexes. That sexual dimorphism, as we mentioned, is like a difference in appearance between males and females within the same species. So that can lead to competition between and among the members of the same sex. And so what this results in is males most often having to have secondary sexual equipment, such as antlers or like sophisticated sexual characteristics for like fanciness, like in many birds, like the peacock feathers here. So we have like the mane of a lion, the coloration of different birds, as well as like the antlers, can all be attributed to trying to entice or encourage or success in mating. All right, that kind of wraps things up for us. We will continue Hardy-Weinberg practice in class the next time we meet.